Alright guys, we're going to be moving on to our next lesson, so I hope DNA went well. This is going to be kind of an expansion, and it's definitely going to have some overlap from your DNA lesson. This particular lesson is going to be all about protein synthesis, which this is a kind of a key essential idea in biology, and it's definitely something we need to have a general understanding. It ties together a lot of things that we've been covering. And so in order to understand protein synthesis, we do need to understand a little bit about the molecule RNA. And so we've just learned pretty much the main information about DNA. We know that it's a polymer of nucleotides, and it has two strands, and it has the nitrogen bases and the sugar phosphate backbone. And your RNA molecule is going to be very, very similar. However, there are going to be three key differences between the DNA molecule and an RNA molecule. And so number one is going to be the sugar that is in the nucleotides. So we still have a polymer of nucleotides. We're still going to have chains of nucleotides. But instead of having the sugar deoxyribose, we're going to see the sugar ribose. And this is where we get the letters in our molecules. DNA was for deoxyribonucleic acid, and RNA is for ribonucleic acid. Now the phosphate group is the same kind of chemical as we saw in DNA. It looks the same. It's going to have the same basic structure, and it's also going to be in the backbone. So just like before, we have a sugar phosphate backbone, and then we do have a nitrogen base on each nucleotide. But here's where our second difference comes in, and that is instead of thymine, we have uracil. And uracil is going to be very similar to thymine. It's similar enough that it can bond to adenine just like thymine did. So if we were looking at RNA molecules, we will not see any T's. Any place where there would be a T, you would see a U instead. And so when we're doing the processes of protein synthesis, a T on the DNA would pair with A on the RNA, and an A on the DNA would pair with a U on the RNA. And so that's going to be an important concept to understand. And then the third difference is that instead of having two strands of nucleotides, RNA is typically single-stranded. Now it can fold up and take on some funky shapes, and we're going to see that with tRNA, but it's usually just one strand of nucleotides. There are three main types of RNA, and really in our lesson today we're going to talk about two of them. We're going to talk about messenger RNA, which is going to serve as a messenger from the nucleus to the ribosome. We're also going to talk about transfer RNA, which transfers amino acids. It's going to be our delivery boy. And then the third type is ribosomal RNA, or rRNA. And this particular RNA type is just part of the structure of the ribosome. I'm sure it does have an active role in protein synthesis, but really this is all we're going to talk about when it comes to ribosomal RNA. We also need to do a quick little review about proteins, because if we're talking about making proteins, we need to go back to our macromolecules lesson and just make sure we understand a little bit about protein structure. So number one, proteins are also a polymer, and they're made up of the monomer amino acids. These amino acids are joined by a bond called a peptide bond to form a chain called a polypeptide. We do have 20 different amino acids that exist, and so each protein is going to be a unique combination of amino acids in a unique sequence. And some of these amino acids we can make in our bodies. Some of these amino acids we have to consume in our diet. You may have heard of the term essential amino acids. But this is one of the reasons why we have to eat protein, is because we have to break proteins down to get the amino acids so that we can build new proteins in our body. So we have 20 different amino acids that are going to be joined up by peptide bonds. And then once we have this polypeptide here, so their basic structure is just this chain of amino acids, a link to amino acid, link to amino acid. And so these lines represent peptide bonds in between the amino acids. And this basic chain here is called a polypeptide. And so in the process of protein synthesis, we're going to be making this polypeptide. But that is not a functional protein. In order to become functional, the amino acid chain is going to twist and fold and glob up, and it's going to go all the way up to at least tertiary or quaternary structure. And this very complexly folded shape is where we have a functional protein. And hopefully something that we learned back in macromolecules is that proteins are always specific. We saw that with enzymes, they only work with a certain substrate. Proteins always have a very specific job because they have a very specific shape. And that shape, all of this twisting and folding, always goes back to this primary structure of the order of the amino acids. And so these proteins are coded for by our DNA. And this is why we say that DNA is the code for life, is because that DNA holds the code for making these proteins in our genes. And so when we're going through the process of protein synthesis, we're not necessarily copying our entire DNA sequences. We're just copying a gene, the gene that codes for a particular protein. And for the most part, one gene is going to code for one protein. Now, in terms of the process of protein synthesis, the proteins are going to be made at the ribosomes. And remember, ribosomes could be out there in the cytoplasm, or they could also be attached to the rough ER. And so the actual process and building of the protein will happen on a ribosome or at a ribosome. Now, we have a small problem here in that DNA, which holds the code, the original blueprint, it's in the nucleus and it can't leave. DNA has the two strands, and so it's quite a bit bigger than RNA, and so DNA cannot leave the nucleus. It's too big. It can't fit through the nuclear pores. And honestly, we wouldn't want it to leave, because if it were to leave the nucleus, it could get damaged or degraded or mutated, and we really don't want that to happen. So DNA is stuck in the nucleus. Now, in terms of protein synthesis, we have to get the instruction to the ribosomes. And this is where RNA is going to come in, specifically mRNA. And RNA is going to act as a copy of our DNA. And that way, kind of like if you have a blueprint for a building, the contractor or the architect is not going to give out the original blueprint. They're going to make copies. And that way, if a copy got damaged or something happened, they could always go back and make another copy. And that's what RNA is going to do for us here, is we'll make a copy of the DNA in the form of RNA, and it will be a disposable copy that will carry the message from the nucleus to the ribosome. And so if we could sum up protein synthesis in one statement, it is this right here. This is called the central dogma of biology, which means it is an essential concept. And if you know anything about protein synthesis, you need to know this statement right here. And that we're going to start with DNA. We're going to go from DNA to RNA. Really, it's going to be mRNA here. And then from the mRNA, we're going to make the proteins. And this is really a summary. Okay, we have two major stages. We have transcription. And transcription is going to be this first stage, DNA to RNA. And then that's going to be followed by translation, which is going to be RNA to proteins. Now, if you ever get these two mixed up, okay, pay attention to the alphabet. Okay, they both start with trans, so we have a C there and an L there. So C comes before L in the alphabet. And so transcription is always going to be the first process, DNA to RNA. And then translation will be the second process, RNA to proteins. And so we're going to take a look at each of these processes in a bit more detail, because of course it's more complicated than that. And so we're going to start with transcription, which is taking the DNA and making an mRNA molecule. Okay, so what we're going to do is we're going to, in the nucleus, because that's where DNA is, we are going to open up the DNA and we're going to make a copy of a gene in the form of RNA. And just like we saw in DNA replication, it's going to be based on this complement system, kind of based on that base pair rule. And this is accomplished by an enzyme that might look a little bit familiar called RNA polymerase. Back when we studied DNA replication, we saw DNA polymerase, and that was our builder. DNA polymerase built DNA, RNA polymerase is going to build RNA. And the actual process of transcription kind of takes place in three basic stages. We have initiation, which is the start, elongation, which is the building of the mRNA, and then termination, which is going to be the stop. And so in the process of initiation, RNA polymerase, there's our enzyme, it's going to bind to a special sequence on the DNA called a promoter. At the beginning of every gene, there is going to be a regulatory sequence called a promoter. And essentially, it's kind of like the starting line for your gene, okay? Because remember, these genes are dispersed all throughout our DNA sequence on our chromosomes. And so when we're going about the process of protein synthesis, we're only going to be copying the gene that we need. We don't copy all the genes all the time. And so these promoters are just little regulatory sequences that pretty much say, hey, RNA polymerase, start here, okay? And so the RNA polymerase is going to bind to that short little regulatory sequence called the promoter. It's going to separate the two DNA strands. And that promoter is also going to tell that RNA polymerase which side. Because remember, RNA is just single-stranded. We're not going to be copying both sides of the DNA, just one side, and that is going to be our gene. Now, once we have the RNA polymerase, it's bound to a promoter, it opens up the DNA. We are now ready to start copying the DNA and making mRNA, and that's what happens in elongation. Now, this is going to look kind of similar to what we saw back in DNA replication, and that RNA polymerase is going to build the mRNA in a 5-to-3 direction, and once again, it's going to be based on that base pair rule. So the only difference here is that we're only copying one side, so we're going to be copying only one strand, and instead of T's, we would have nucleotides with U's. So once again, A would go to U, T would go to A, C to G, G to C, okay? And so that's how we go about the process of elongation. That RNA polymerase is just going to add one nucleotide at a time, and what we're producing is what's called the primary transcript or the pre-mRNA. So this is where we build our mRNA, our messenger. Now, how does it know where to stop? Well, kind of like the promoter up here is a regulatory sequence, there's another regulatory sequence called a terminator, which signifies the end of the gene. And so once RNA polymerase reaches that terminator sequence, it releases that mRNA molecule, and the DNA will wind back up and go back into the double helix. And so here's kind of what it basically looks like. RNA polymerase will bind to the promoter, it will copy one side, it will build the new strand 5-to-3, and it will stop when it reaches a terminator. Now, we're not technically ready to go yet because we have to do a little bit of editing to our mRNA. It's kind of like when you edit a paper for English. Sometimes you have to make some corrections, or sometimes you have to go back and maybe reword or cut some things out. And that's essentially what we're going to do with our mRNA. So one of the issues is that within our DNA, we have lots of sequences that technically don't code for anything. Now, they used to refer to these as junk DNA, but some of them have functions, some of them they still don't know what they're for. But all throughout the DNA genome, there are these pieces that do not code for proteins, and they're interspersed even within the genes. And so these pieces that do not code are called introns. And so I like to remember the junk in the trunk. You got the N here. And so the introns are the junk DNA that do not code for our protein. And then we have the exons that are expressed. These are the ones that we need. And so I also like to compare this like to the note sheet that you're writing on right now. If you were to go to a college textbook and you were to crack it open and read the section about protein synthesis, it would be pages and pages and pages. Okay, I would say it would probably be at minimum at least a good 6 to 10 pages just on the lesson that we're learning right now. Now, obviously, the note handout in front of you is a shorter copy than that. Because in the textbook, you have things that you don't need. You have little captions, you've got stories, you've got intro paragraphs and summaries at the end. And so that's kind of like our DNA. When we copy the gene, it's like copying the whole chapter. But in reality, I don't need all of it. So I can cut out the pieces that I don't need. In this case, it's the introns. And then I put back together the pieces that I do need, and that's going to be the exons. And so that what I have in the end is our final copy, our final transcript. And it's kind of like the note sheet that you're writing on now. That is the information that you need to know for the test, okay? I cut out all the stuff that you didn't need and put together the stuff that you did need. And now you have just a sheet of important information. And that's what's going to happen to our mRNA. Now, in addition to that, we're going to put some protective end caps. We have a 5' cap, which is a little guanine up here. And then we have a poly A tail, which is several adenine nucleotides. And what these little sequences do is, number one, they help protect the mRNA. It kind of protects it from enzymes out in the cytoplasm. Number two, it helps it get out of the nucleus. And number three, it helps that RNA know where to bind to the ribosome. And so now that we have our edited DNA, we have cut out our introns and put back together our exons. We have added these little protective caps on either end. And now we're ready to go and build our protein. Now, before we can do that, though, you do need to understand how the genetic code works. And hopefully you remember this at least a little bit from freshman year. This is normally something the students kind of like, and they like using the codon chart. But our genetic code is read 3 letters at a time. It's kind of like taking our alphabet. We have 26 letters to the alphabet, and by itself it really doesn't mean much. But if we start putting letters together in certain combinations, they then have meaning. So like DOG, automatically you're probably picturing a dog. CAT, automatically you're probably picturing a cat. And so it's important that we understand that we have a 4-letter alphabet when it comes to our genetic code, but we put them together in 3-letter combinations called codons. And these codons are on the mRNA molecule. So the DNA was first, mRNA is made, and we're going to read that mRNA in these 3-letter sequences called codons. And what the codons code for are amino acids. Now, there are a few that code for a stop, which signifies the end of a polypeptide. But there are 64 different codons if you take that there are 4 letters and 3-letter combos. And notice we only have 20 amino acids. So what that means is we could have multiple codons for the same amino acid. And we'll see that as we go through some protein synthesis practice. Now there's a few special codons. We have AUG, which is the start codon. Now it does code for an amino acid, methionine. But essentially all of your polypeptides are going to start with AUG, which means the methionine amino acid is going to be the beginning of each of your proteins. And then we have 3 codons that these don't code for amino acids, but instead they code for a stop, which pretty much tells that the ribosome where to stop reading that mRNA. And so we've got UAA, UAG, and UGA. Now I have included a copy of this in your notes. If you look at the very back page, there's kind of a mini version. You have both the box type as well as the kind of circle type. And I'm not going to take the time to go over how to read it right now. I'll do a very, very brief one, but this is something you're going to want to practice with and make sure you understand how to use it. So if you have the rectangular version, notice that the first letter of the codon is over here on the left, the second letter is on the top, and the third letter is over here on the right. And so let's just very quickly look up our start codon, A. Okay, so A is going to tell me that it's in this row. U is my second letter. Okay, that's going to narrow it down to this box right here. And then I come over and find my third letter, G, and notice it's my methionine or start. Now the one you have in your notes doesn't have these little cheats right here. It doesn't have the 3 letters already put together. So just make sure that you're reading it correctly and that AUG equals methionine, okay? Now the way this one works is we have first letter, second letter, third letter. And so you start from the inside and work your way out. So A, U, and then G, and that is once again methionine. So these have the exact same information on them. Here we have the names of the amino acids. Here we have abbreviations, and it really just depends on which chart you're looking at. But you do need to know how to read a codon chart. And so something else we need to understand is that the genetic code is pretty much universal. Whether you're talking about a bacteria or a fruit fly or a hermit crab or a human being, okay? Pretty much the way the code works is the same for every organism. We have the mRNA codons that code for amino acids. That AUG is going to be methionine for pretty much every species. And so the whole idea of the genetic code being universal does show that living things are connected in some way, okay? All right, and now let's go ahead and talk about the final stage of protein synthesis, which is translation. And once again, this is going to be the second half of our central dogma. This is where we're going to take the RNA and we're going to build our protein. So because mRNA is skinnier, it only has 1 strand, it can leave the nucleus, and it's going to go out into the cytoplasm or to a ribosome on the rough ER, and it's going to attach to a ribosome. Now, the ribosome is going to move along the mRNA, and it's going to read it 3 letters at a time. And then we're going to have another mRNA or another RNA molecule called tRNA is going to be our delivery boy. And so you can see over here that tRNA kind of has a unique shape. It kind of folds to form this little looped structure. It's still only one strand of nucleotides. It's just folded up. But on one end, we're going to have the amino acid that attaches, and on the other end, we're going to have a 3-letter sequence called an anticodon. And this is going to be a check mechanism to make sure that the right amino acid is being brought to the right place. So the mRNA codon determines the amino acid. The anticodon is just going to be a check mechanism. And so just a little bit more on tRNA. So as I mentioned earlier, it kind of folds to have this unique looped structure. On the 3' end is where we have the amino acid attachment site. And whichever amino acid is going to be attached here is going to be determined by that codon. So if this was my anticodon, then the codon would have been UUC, okay? And so that would determine a particular amino acid that would attach right up here. And so these tRNAs become charged, or they bind to those amino acids out in the cytoplasm. The amino acids are already out there, the tRNAs are already out there. Remember that this is just a chemical thing. And so in terms of the ribosome, we kind of need to understand a little bit more about it. So ribosomes have these 2 subunits. They have a small subunit where the mRNA binds, and then they have a larger subunit. And within the ribosome, we have 3 important sites for the tRNAs. We have the A site where the incoming amino acid arrives. We have the P site that will always hold the growing polypeptide. And then we have the E site where the tRNAs are going to exit. And so like in our pictures here, the tRNAs are always coming into the A site. They're then going to shift over to the P site, and that's going to be the one that has the growing polypeptide chain. And then from the P site, they're going to shift over to the E site, and they're going to exit and go pick up more amino acids. And so kind of like we saw with transcription, we have 3 stages. Once again, we have initiation, elongation, termination. This time, initiation begins with that start codon. So that very first amino acid, methionine, is going to be brought by tRNA into the A site. It'll then scoot over to the P site. Then the next amino acid will be brought by the next tRNA, dependent upon the code. And these amino acids will be brought in one by one. Now, whenever the amino acids are in the A and the P site, so they're attached to their tRNAs, it is when we have those 2 amino acids side by side that a peptide bond is formed between the 2. And then once the P site moves over to the E site, that tRNA exits, and that growing polypeptide is always going to be right here on the P site, okay? Now, this will continue 1 amino acid at a time until a stop codon is reached. And that signifies the end of our polypeptide, and at that point, the polypeptide is going to be released. And so here we have our incoming amino acids coming into the A site. Notice I have my growing polypeptide on my P site, and then my tRNAs are exiting, and that ribosome is going to move all the way down that mRNA until it reaches a stop codon. Now, the reality is it's not like the mRNA gets read just one time and it's over. You could have many ribosomes reading that mRNA at the same time. You could have that mRNA being used by multiple ribosomes, and pretty much as long as that mRNA is around, it can be translated into a polypeptide. Now, there's lots of chemical signaling that's going to be going on. We're not going to want to make things that we don't need. And so once we get enough of that particular protein, there will be chemical signals that pretty much say, hey, we're done with that mRNA, and eventually that mRNA will be degraded, and that's the point at which we stop making that particular protein. Now, once translation is complete and we've made our polypeptides, remember that this is not a functional protein. This is just my basic amino acid chain. It has to twist and fold into this complex shape, and so once it takes on that 3D shape at the quaternary or tertiary level, that is the point at which we have a functional protein. And then the last thing we're going to cover are gene mutations. Now, back when we covered genetics, we did kind of focus more on chromosomal mutations. We mentioned these, but these are going to be what we call point mutations, where you have individual nucleotides that are essentially not right, okay? We could have a substitution, where maybe like in the picture up here on a substitution, originally there was a C, but instead there was a T. Now, the thing with the substitution is at most, it should only affect one amino acid. Okay, that doesn't mean they're good. They could still be really, really bad. It all depends on where and what the change actually causes. Sometimes that change could be silent, which means it actually doesn't change the amino acid at all. Remember that multiple codons can code for the same amino acid, so it possibly could have no effect. It could lead to just a change in one amino acid. And that may not sound like a big deal, but if just even one amino acid changes, that's going to change the folding of that protein. And so if it doesn't have the right shape, it's probably not going to function correctly. And then a third thing that could happen is it could actually change a codon to a stop, and it could cause the protein or the polypeptide to be stopped too early. So it really just depends on where and what, but at most, a substitution would change one amino acid. Now the other types, a deletion or insertions, if we added a nucleotide or two or took out a nucleotide or two, it actually affects the rest of your polypeptide because it causes what's called a frame shift. We're always going to read the letters in groups of 3. And so like in the case here where 1 was added, it completely shifts the reading of the 3s. So now instead of CTT, we have CCT and then TCT. And that's going to keep on going all the way down the chain, and that's going to result in potentially an amino acid change everywhere else after that mutation. And the same thing would happen with a deletion. It frame shifts and changes in the readings of our 3s. And so essentially, any mutation could be a bad mutation. Sometimes mutations are good. It really just depends on where and what. But in the case of gene mutations, we just have individual nucleotides that are incorrect. And at some point, you guys are going to practice that. We probably won't get that in before the Thanksgiving break. Also, you will have a quiz over protein synthesis, but it won't be until after the break. I don't want you guys to have to study or do anything over the break, and so we'll do a review and do an activity or something when you come back. I do want you to hopefully take advantage of the review activities that you're doing before the break. And if you guys have any questions or anything you need help with, feel free to reach out. I hope you guys have a wonderful break, and I will talk to you soon.